Hi, my name is Karin Gillner. I'm one out of 3 project coordinators at the National Genomics Infrastructure, or NGI as we're called as well, in Stockholm. And I'll give you a very brief introduction to what we offer here in terms of applications and instrumentation and also bioinformatics support. And after my talk, you will hear more in-depth talks about the bioinformatics support that we supply. And NGI Sweden is the largest facility at SciLife Lab. We started 2013 and are divided in 2 nodes in Stockholm as well as Uppsala. And we have received funding from the Swedish Research Council, the universities involved, as well as SciLife Lab. And this means for you as a user that we only charge you for reagents as well as plasticware, etc. And our mission is to make a state-of-the-art infrastructure for massively parallel DNA sequencing and SNP genotyping. And this should be available to all researchers throughout Sweden. And we are 3 nodes, in fact, 2 in Uppsala and 1 in Stockholm. The one in Stockholm is directed by Joachim Lundeberg, and we have Ulf Gyllensten at the Uppsala Genome Center and Ann-Kristin Syvänen at the SNP and Seq technology platform. And perhaps what differs between us and other facilities in the world is actually our support that we're able to give in terms of planning your projects and support during conducting your experiments and the methodology that we supply here, as well as that you will hear more about afterwards, and the bioinformatics support that you can receive from us. And the 3 nodes, the 2 nodes in Uppsala and the Stockholm node, we all share a common order portal where you not only place your orders, but you can also find a lot of information there in terms of the applications we supply, but also documents with, for instance, sample criteria and price information. We are 6 project coordinators in total, 3 here in Stockholm and 3 in Uppsala. And we sit down 2 times a week to divide the projects between us based on the sequencing technology required and also the application in question. And we also take into account the queue situation, as well as, of course, the geographical position of the group in question, to make it as efficient and easy for you as a user as possible. And if you go back and only talk about us here in Stockholm, the NGI Stockholm node, we are divided into 2 functions or groups, you could say. We have the productions group, which currently is headed by Karsten Daub, while Ellen Sjöver is on maternity leave. And here, all standardized and validated protocols, instruments, and data analysis tools will be run. And based on the communication we have with your users and needs that we pick up, might be technologies which are upcoming and that you as a user find of interest. Then we have the applications and development function headed by Max Keller. And here they will work on library preparation protocols, new instrumentations, and also build new data analysis tools. And the goal is to have all these validated and later transferred into productions. So we have a synergy trying to constantly improve our methods and developing new tools to always be in the front line of the technologies available. The instrumentation that we have in Stockholm is the Illumina sequencing instruments. We have the HiSeq 2500, where most RNA-seq projects, for instance, are run. We have the MySeq systems for smaller genomes, and it's also good for a little bit longer reads. We will shortly have the new NovaSeq 6000, which basically will replace the HiSeq 2500 instruments. And we also have the HiSeq X, where we run the whole genome sequencing projects on. And since this talk will focus mostly about what's novel, I'll mention a little bit about the new NovaSeq 6000, which we in fact will receive this very week. And this instrument will give you up to 1000 gigabases per flow cell, compared to, for instance, the HiSeq X, where you can get up to 100 gigabases. And that we used to call a lot of data. So this is 10 times more than that. And you'll be able to have a read length between 2 times 50 base pairs up to 2 times 150 base pairs. And the runtime will also be significantly shorter. For instance, if you would sequence 50 million reads per sample, you could fit 132 RNA-seq samples in one such flow cell, or 16 human genomes. What you as a user might be most interested in is the price, of course. Since this is such a new instrument, we don't have an exact estimate yet, but we believe it will end up in about 50% of the cost per base for the same compared to one base on the high output on 2500. So about a 50% reduction in the cost per base. And the other thing which is new is that Illumina has released the HiSeq X, where we run whole genome sequencing on. So now it's released for not only the human genome, but all other genomes as well. And you can sequence down to 15x as compared to 30x, which has been the limit before. And as I mentioned, one lane here gives you 100 of gigabases of data, which means about 30x coverage for the human genome. And you'll get this for about 9,000 kronor. Other instruments available in Uppsala is also the PacBio system, where you can get long reads, and you also have the ion proton and ion PGM systems there. Our key applications at productions in Stockholm are the following: RNA-seq, whole genome sequencing, de novo sequencing, and cancer whole genome sequencing, or normal tumor per whole genome sequencing. And these are the 4 applications that you will hear more about in terms of the bioinformatics side later on. The library preparation methods we have at productions for DNA are the following. We have the standard library preparation method, which is the TrueSeq DNA PCR-free. We also have 2 low input methods, Rubicon ThroughPlex, for instance for ChIP-seq material, and we also have Lucigen, which is not yet validated, but it will shortly be validated. For targeted enrichment, for human whole genome exome sequencing, we have the Agilent SureSelect XT preparation. And for mate pair library preparation, we have libraries up to 20 kb in size. However, the largest, the 20 kb, is not yet validated. For RNA, we supply ribosomal depletion and poly-A selection. And for small RNA, the TrueSeq small RNA library preparation. If we look at what application and development can do for you, and methods that we hope will be validated and transferred into productions eventually, they are working on low input protocols for RNA-seq, as well as for small RNA-seq. There are new technologies for long reads, such as the 10x Genomics Chromium and also the MinION system, which can sequence up to hundreds of kb, and it's also the only portable system. We have protocols for cost-efficient library preparation, such as RADseq and Nextera User QC, which basically means that you supply us with an exact amount, an exact volume of material, and we don't do any QC of the samples. We also have epigenetics library preparation protocols, ATAC-seq, and also bisulfite sequencing. One of the most common questions that we get is about the turnaround time. And if we divide it up into protocols or projects where we are actually producing the libraries and projects where we only do sequencing, there is of course a difference in time. But for library preparation projects, 50% of the projects are actually finished within 5 to 8 weeks. Another 30% will finish within 9 to 12 weeks. And we do actually have projects around 10% that are finished within the first 4 weeks. So this means that 90% of all projects will be finished within 9 to 12 weeks, within 12 weeks. For sequencing only projects, more than 70% will be finished within 4 weeks. And another 20-25% within 5 to 8 weeks. Thank you for listening, and I'll now hand over to our bioinformatics crew, who will talk more about the main applications.